What if I told you? Scientists have learned to edit the very code that makes you, you. What if I told you that deep within every cell of your body, there exists a book, a biological manuscript, billions of letters long, containing the complete instructions for building and operating you? This is your genome, the very code that makes you, you. For all of human history, this text has been immutable, a story written in stone, passed down through generations, unchangeable. But that is no longer true. We are living at the dawn of a new era, an age where scientists have finally learned to become editors of life itself. The letters that decide your face, your height, your intelligence, even your predisposition to certain diseases, can now be changed like text in a document. Imagine a single typo in this vast genetic library, a tiny error that could lead to a lifetime of illness. Now, imagine having the power to find that specific letter among billions and correct it. This isn't science fiction. It's like having a biological find and replace function for our own DNA. A way to press Ctrl+Z on evolution itself. This revolutionary technology allows us to correct genetic mistakes, enhance desirable traits, and even rewrite the future of our species. It's called CRISPR, and it's arguably the most powerful tool humanity has ever built. A pair of molecular scissors, guided with unprecedented precision, that can cut, paste, and rewrite the source code of any living organism. The potential is breathtaking, curing genetic diseases like sickle cell anemia and Huntington's, creating crops that can withstand climate change, and even eradicating pests that carry deadly illnesses. But with great power comes a question no one's ready to answer. We stand at a monumental crossroads, holding the keys to our own evolutionary destiny. The lines between healing and enhancement are blurring. The ability to alter the human germline means changes we make today could echo through all subsequent generations, forever. We have unlocked a force of nature. So, what happens when we reprogram life itself? The story of this revolution begins not in a human cell, but in the microscopic world of bacteria. For billions of years, bacteria have been locked in an endless war with viruses. To survive, they evolved a sophisticated immune system. In 2012, two scientists, Jennifer Doudna and Emmanuel Charpentier, uncovered the secrets of this ancient defense mechanism. They realized that what bacteria use to fight viruses could be repurposed into a revolutionary tool, one that would give humanity an unprecedented power. They had found a way to edit the very code of life. This system is called CRISPR. Think of it as a genetic library of past infections. When a virus attacks, the bacterium saves a small piece of the invader's DNA in its own genetic code. This acts as a memory. If that virus or a similar one ever returns, the bacterium is ready. It creates a special molecule, a kind of genetic bloodhound, called a guide RNA. This guide RNA carries the signature of the enemy virus and hunts for a match within the cell. But the guide RNA doesn't work alone. It partners with a powerful protein, an enforcer called Cas9. Together, they form a search and destroy unit. The guide RNA leads the Cas9 protein through the cell's genetic material, scanning for the exact sequence it was programmed to find. Once it locks onto the target, the invading viral DNA, the Cas9 protein activates. This is where the magic happens. The Cas9 protein acts like a pair of molecular scissors, making a clean, precise cut through the DNA, disabling the virus and saving the bacterium. Doudna and Charpentier's breakthrough was realizing they could create their own custom guide RNA to target gene in organism. Suddenly, this bacterial defense system became a universal tool for gene editing. It's the biological equivalent of a find and replace function in a word processor. We can design a guide RNA to find a specific faulty gene, like one causing a genetic disease, and use Cas9 to snip it out. The cell's natural repair mechanisms then kick in, often using a healthy template we provide to paste in the correct sequence. With this technology, genetic diseases that have haunted families for generations, conditions like sickle cell anemia, certain forms of muscular dystrophy, even some types of inherited blindness, were no longer an immutable fate. They became, in principle, editable. The implications are staggering. For the first time in the 4-billion-year history of life on Earth, evolution was no longer entirely random. It was becoming programmable. CRISPR didn't stop in the lab. By 2020, doctors had already cured real patients using gene editing. A woman who was blind from birth could now see light for the first time. But scientists weren't satisfied. They asked, if we can fix diseases, what else can we change? Height? Memory? Intelligence? The line between medicine and modification began to blur. In late 2018, the scientific world was rocked by a secret that had been kept for months. A Chinese scientist, He Jiankui, announced he had done the unthinkable. He had secretly edited the genes of twin baby girls. His goal was to alter a gene called CCR5, theoretically granting them resistance to HIV, the virus their father carried. But in doing so, he didn't just edit two individuals. He edited the human story itself. To understand the gravity of this, we need to know the difference between two types of gene editing. Somatic editing is like correcting a typo in a single copy of a book. It affects only the patient, and the changes die with them. But what He Jiankui performed was germline editing. This is like changing the master printing plate. The edits, made to an embryo, are permanent and heritable. They are passed down through every subsequent generation, forever altering a family's genetic lineage. When the world found out, it exploded. The condemnation from the global scientific community was swift and nearly unanimous. Emergency summits were called. Leading researchers who had pioneered CRISPR technology expressed their horror. He was called reckless, unethical, a mad scientist who had broken a sacred trust. The experiment was not only dangerous, with unknown risks of off-target mutations, but it was also medically unnecessary. Proven methods already exist to prevent HIV transmission from parent to child. This wasn't about curing a disease. This was about crossing a forbidden frontier. This was redesigning humanity. For the first time in history, human beings were born with DNA that had been intentionally and permanently altered in a lab. The experiment was immediately banned. The scientist was condemned and eventually jailed. But the line had already been crossed. The theoretical had become real. Pandora's box was open, and there was no going back. The world now had to confront the reality that the power to rewrite our own species was here. And the question was no longer we could do it, but we would control it. Still, CRISPR isn't evil. In fact, it's saving lives every single day. Doctors are using it to kill cancer cells by rewriting their DNA. Farmers are editing crops to survive heat and drought. Scientists are trying to eliminate malaria by editing mosquitoes. And now, CRISPR 2.0 can even edit one letter out of 3 billion, with perfect precision. Humanity has gained the power of a creator. But our wisdom hasn't caught up yet. But what if someone uses CRISPR not to heal, but to control? A single edit could create super-resistant viruses. A mistake could cause genetic mutations that last for generations. And here's the scariest part. Once you edit a human embryo, that change passes down to every future child. You're not just rewriting one person, you're rewriting the future of the species. So now, scientists face a question. Just because we can edit life, should we? Where is the line between cure and creation? If the rich can afford to make perfect children, taller, smarter, stronger, does that mean natural humans become obsolete? We could soon live in a world where biology decides destiny, not effort or talent. A world where evolution is not natural, but engineered. CRISPR is humanity's most powerful invention, and its most dangerous. We're rewriting the story of life, one gene at a time. But the question remains, when we finally hold the pen that writes the next chapter of evolution, will we write a miracle or a mistake? This is Carl, and you've just watched Quantum Queasy. Subscribe, because the next video might just change the way you see life itself.